So we are going to be doing a lab where we're going to measure the rate of cellular respiration in an organism. And the organism in which we are going to measure this is in seeds. And so I have 3 different kinds of seeds. I have, let me close this bag up so I don't spill all over the place. I have Alaskan peas, I have mung beans, and I have rye grass, all right, grass seed. And so then we're going to measure the rate of respiration here. These seeds, let me just get some background information on seeds. These seeds here, are they alive?
No.
How many people think yes? How many people think no?
That's what happened.
There is a lot of people that don't have a thought about this. they're not so sure about this. All right, so some people, I saw a lot for the no, like this kind of thing, all right, on here. These seeds, do you think that we'd be able to plant them and get a plant?
Can I change my answer?
Yes. Sure.
All right, I'm going with yes there.
All right. These, eventually, if we planted these, they could grow into a pea plant or a bean plant, or the rye seeds would be in a ryegrass. All right, and so therefore, to grow into something living, these guys must be living. All right, so let's talk a little bit about a seed. Does anybody want to take a chance and raise their hand and tell me what is a seed? Like, what does it contain? What is it? What is a seed? Nobody? It's been very popular. Not a lot of people have known a lot about seeds all day long, so you're in good company. Yes.
It's a plant egg.
All right, it's a plant egg. All right, it's a plant egg. Anybody else have something? No? Nobody's got anything? All right, let me talk about how a seed develops to give you some background information. So a seed develops from a flowering plant. And so when we look at, let's talk about just an apple seed, something that you're familiar with. I'm going to attempt to draw an apple blossom. All right, so just bear with me. All right, so, and I'm going to draw the structures that are important for this. So in the middle of a flower, you have the female part that kind of has this shape here. And this female part is surrounded by male parts of the flower. So these male parts of the flower stick up, and there's usually a bunch of these. And then this is in the center of your flower, and then surrounding it, this is the bear with me part, you have your petals. All right, so guess you know what I'm talking about in the middle of the plant? You have the center part with the other stuff. Okay, so you're following me here. So this part that's sticking up right here is the male part of the flower. The male part has on the very top pollen. You guys have heard of pollen, right?
Yes.
Do you guys? Thank you for answering. So pollen. Does anybody know what pollen contains, or what it is? This is the male part of the flower, right? Inside of this pollen grain is actually a sperm, all right? It's the sperm of the flower. Now, that's the male part, and the flowers have both the male part and the female part. So now in the female part here, if we go down this base, it's called the ovary. And inside the ovary, there are little compartments, they call them ovules. So I'm going to draw a few compartments here. And inside each one of these ovules, what do you think is inside? It's the female part. This is the egg. So this is where your eggs are. Inside, at the bottom of this ovary in these little compartments. So now what happens is, so this, if you're writing this down, this is an egg. This is the pollen. Let's label this. Pollen, remember, contains the sperm. When I put parentheses, it contains the sperm. Pollen and sperm are not the same thing, but contains the sperm. So then the thing with flowers is that they can self-pollinate, so the pollen from the same plant can fertilize eggs of the same plant, or that pollen can come from a different flower that's called cross-pollination. And so therefore you can take pollen from a totally different plant and put it on here. So then what happens with the pollen? Let's do a self-pollination here. So the pollen from that same plant lands on the top of the female part here. And what happens is that the sperm inside of there, from the pollen, they grow what's called a pollen tube. It's a literal tube that goes and ends at one of these compartments with the egg. And so therefore the sperm moves down here and goes to the egg, and you get the sperm fertilizing the egg. You get a one-cell little baby plant that's called a zygote, and then it starts to divide by mitosis and grow into a little baby plant. So that pollen moves, that sperm moves down here, and eventually, I'm going to change my color here, you get, I'll put a little baby plant in there. So then the next pollen that comes, either from this plant or it could be a totally different plant, the next pollen lands here, grows a pollen tube. So let's say, I'll go through an example of this one here. So it grows another pollen tube. And let's say it goes to this ovule. And so then the sperm fertilizes this egg. And now we get another little baby plant in this little compartment. And this can continue to happen until all the eggs are fertilized and you get little baby plants. And so, so then what happens is you've got these little baby plants, and the outside of the ovule here develops a really tough outer coat. This develops a really tough outer coat here. And if these happen to do the same thing, these guys would develop, if these guys were fertilized as well, you get all of these tough outer coats. Then what happens, that's your seed. And so what happens is then the flower dies off. And guess what the ovary does? The ovary in a flowering plant develops into the fruit. So, and then you have the seeds in the middle. So if this was an apple blossom, the apple that you eat, the fruit that you bite into, is actually the ovary of the flower that it was once came from. All right? All right?
So
It's a whole different spin on eating apples. Right? So, yes.
I was right.
Oh, kind of. Fertilized egg cell. They grew. And so on. All right. So when you cut open the apple and you see those different seeds, these are the little seeds. All right. So then when we ask, or going back to that question, what is a seed? If I enlarge a seed here, you have your little baby plant inside your seed. You have the tough seed coat, so that's why these seeds here are hard. And you have the inside here, you have this part all here. This is called the endosperm. This is food for the plant. And so that's what's in there. So those are the main three things of the seed. The food, the baby plant, and the tough seed coat. And so what happens is in these peas are not growing right now. So they are what we call in a dormant state. They are doing just enough to survive. Inside the seed, the little baby plant, can it do photosynthesis inside the seed? No, absolutely no sunlight getting in there. All right, so no photosynthesis going on. It is alive, so it does need some energy. So it does go through cellular respiration. The glucose for cellular respiration comes from the endosperm, but they're doing so little, just the bare minimum to stay alive. So that's what we have here in these dry seeds. Now what I'm going to do is I'm going to try and get these seeds out of dormancy. So what do you think a seed needs to become not dormant and therefore start growing and getting these little baby plants to grow out of these seeds? So what do they need? Raise your hand if you have an idea. Now that I asked you to raise your hand, not to say.
Seriously.
Just a minute ago, everybody was mumbling. What's a big thing that you need for any kind of plant to grow? Water. Huge. So water. So H2O. All right. H2O is one thing, and that's one of the main things from the environment. So what I'm going to do is overnight, so I'm separating these guys out into beakers and so on. I'm going to fill this up with water, so it's soaking in water, and by tomorrow, that water will absorb into the seeds, and tomorrow morning, these seeds will probably be up to here, because they will have soaked up the volume of the water. That's going to trigger it to start germinating. Then what I'm going to do is I have these cookie trays. I bring in all my cookie trays from home, and spread out the seeds. I put some wet paper towel on the bottom, spread the seeds out, and put wet paper towel at the top, and so it keeps them moist. All right, and then I keep them inside. It's warm inside, so that's another thing. Is warm. The temperature is another thing that triggers. So my seeds will not begin to break out of dormancy unless it's warm outside. So if I stuck them outside, they would not happen. This is so that the seed is not going to start to sprout, and the baby is not going to start to grow unless the environmental conditions are right. So the environmental conditions, you need to have water for the plant to grow. The temperature needs to be the right temperature, and so on. So then this will trigger this plant to come out of dormancy, and then it'll start to grow. And so when it grows, it's going to grow in two different directions. It's going to grow through the roots and through the stem. Anybody know which one pops out of the seed first? The root or the stem? How many people think it's the stem? Okay. How many people think it's the root? All right, it is the root. The root comes out first. What does the root do? Takes in the water, right? And so the root, oftentimes you'll see, we'll have to see how these, oftentimes as the root sprouts out, sometimes we can see little root hairs on it. What do the root hairs do? Increase surface area so that the root can take in more water, right? And so let's talk about why this happens. So why the root comes out first. Usually when you plant a seed, this is the ground, you plant them underneath the ground. So here's your seed and your little baby plant inside. And I'll just do it all in black here. So the little baby plant, and then if the temperature is proper and it has enough water, the root will come out first, and then eventually it'll sprout out and keep on growing until eventually it pops open above the ground. So what's going on is while it's still underneath the ground here, the water, the root comes out to get water, but the plant has energy still to grow from the endosperm in it. So therefore it doesn't, it gets all of its energy to grow here, and then it's taking in the water through the roots, and so then once it eventually breaks the ground, now it can do what? Photosynthesis, because the sunlight then hits it and it can make its own glucose and it won't be dependent on the endosperm. And so that's why the root comes out first, because you don't have any other water source besides what's in the ground. The leaves are for photosynthesis. It has a store of food there for the growing plant until it reaches the ground here. It breaks open and gets to the sunlight. This is also why, if you look at the back of seed packets, they'll tell you how deep to measure or to plant the seeds. This is because this has to do with the amount of endosperm that you have here. So therefore, if you plant a seed too deep in the soil and this little guy starts to grow, it's going to keep on growing and growing and growing. But it may run out of endosperm and therefore energy to get ATP to go through cellular respiration before it ever hits the ground. And therefore, it's going to stop growing because it has no more energy because you used up all the endosperm. But it can't do photosynthesis yet because it hasn't broken the ground, and it'll die off. And so sometimes seeds, you plant them too deep, you'll say, oh, they didn't sprout up, and that's usually why. Okay? And so that's your background about seeds. Now the seed itself, in this state, all right, and when I put them in water and so on, the seed we just established is not going through photosynthesis. It just goes through cellular respiration. So the seed is a good specimen to be able to be used to measure the cellular respiration because we can control the seeds, they're not moving around and so on. One year I tried to do this with crickets and it did not work out so well. All right, we had a lot of cricket deaths and a lot of things going on, and so I'm going to stick with the seeds. I gave it a shot one year, but the seeds are easily manageable. They're not moving and so on, and they're not doing photosynthesis. This is why we use seeds instead of a little baby plant, because a little baby plant you'd have both photosynthesis and cellular respiration going on, and so you'd have two processes, and it'd be hard to measure the rate of cellular respiration versus photosynthesis. All right, so that's our background on seeds. So we are going to be using seeds. So then let's look at the equation. So then in your packet, I talked about the equation for cellular respiration. And we are going to be measuring the rate of cellular respiration. So that is how much cellular respiration is occurring over time. So like per minute or something like that. So when we're looking at this, when you're trying to measure a process, there are different ways that you can measure. And so it talked about how up here that you could measure how much of the reactants it took in. So you could measure how much oxygen the seed took in. Or you could measure how much CO2 was produced, or how much water was produced, or energy was produced. And so in this lab, what did it say that we are going to measure in the reading? It said we were going to measure the rate of cellular respiration by measuring one of those things. Which is it? It's on the first page, so you could actually look. Towards the bottom. Got there? What are we measuring? What's happening? The volume of what? Like, what are we going to be looking at?
Changes in pressure.
Right, due to what?
As oxygen is consumed by germinating plant seeds.
That's right. So this is what we are going to be measuring here. All right, and so we are going to be looking at how much oxygen is consumed by the seeds, and therefore how much oxygen, the more oxygen is consumed, the greater the amount of cellular respiration is going on. So that's what we're going to be looking at. So how are we going to be able to tell with the seeds how much oxygen they're taking in? So in comes the use of a respirometer. So I'm going to give you some background information here about the respirometer. This is what it looks like. And so you have not that fancy here, it's just a glass vial. I glued a washer to the bottom because we are going to actually put this into water and it needs to be weighted down, so as a washer. And as a rubber stopper here, through the stopper is a hole in which there's a glass pipette. It measures volume, it measures milliliters, and I put some epoxy here to make it so that there's a nice clean seal. So the only entrance into the respirometer on the bottom is through the tip of the pipette. All right, so that's what that looks like. So I'm going to write on the use the projector here and talk and draw a little bit about the respirometer. So I'm going to look at what we're going to do. So here we have the couple side. So let's draw this together. What we're going to do. So the respirometer looks like this. So let's go ahead and draw it here where this is the washer right at the bottom that I just showed you. And here is the pipette, and the stopper. There's your stopper, here's your pipette, and so on. It tells you in the reading that in the respirometer we're going to use a chemical. Can anybody recall what the chemical is called? What is the chemical? Spencer, do you have your erased or no? No. No. Anybody? Stephanie? Potassium hydroxide. And that is KOH. The K is the potassium, OH is the hydroxide. So what we're going to do in creating this respirometer is we're going to, I have cotton balls, and we're going to use, in this bottom part, absorbent, ABS, I'm going to abbreviate for absorbent cotton. So cotton by nature is absorbent, and so the KOH is going to be in liquid form. So what we can do is actually, without the dropper, we can put drops of KOH. So I'm just going to put here KOH is going to be in the cotton. Now KOH, you do not want to touch your living organisms. So therefore, we have to have a barrier between them. And so we're going to put a layer of, I ordered some non-absorbent cotton. So cotton by nature is absorbent. So what the manufacturer did is put a hydrophobic substance on it to make it so that it's going to like a waterproof substance on the cotton. So it has the feel and look of regular cotton, but liquids will not absorb into it. So why that's important is because cotton is fibrous. It still is going to allow gas exchange to occur, so gas can move through the cotton, but the liquid KOH will not. And so it's going to act as a barrier. And then we can put our organisms, which are the seeds. So these guys are your seeds. Here. So that's a setup of a respirometer. And so what we want to do is measure the rate of cellular respiration, how much cellular respiration is occurring in these seeds. So what we do with the respirometers next is we put them in a water bath. So I have these big flat trays that I will fill with water, and they're going to be setting on the flat part. Here's 3 trays can fit on each pod. So this tray is going to be filled with water, and what you can do is lay your respirometers in there. So let's look at that. Here's my big tray. All right, filled with water. And so you can lay your respirometers in there, and so I'm going to go ahead and dry. Here's the washer. Here's the system. This, and then here you have your pipette sticking out like this. Here's the little tip. All right, so in here you have that KOH. You have your non-absorbent cotton as a barrier, and now you have your seeds. Now, remember I said I had that epoxy and we have the rubber stopper, so we want the only place for anything to go into the respirometer at the end of this pipette. And so, so when you lay these in the water, is water going to go into the pipette?
Yes.
It will go into the pipette. The question is, will it go all the way to the seeds? Think about what's in the respirometer. Grace, what do you think? Air. You have gas molecules in the respirometer. So when you set this up here and you have your 2 layers of cotton and then you have your seeds in there, this is not empty space up here. There's gas molecules in there. And so what's going to happen is that water will move in, but it won't move in all the way. Why? Because there's air in there. Why will it stop? So, at what point will it stop? What's going to make it stop?
Where the air stops on the other side of the water.
Yeah, where the air stops over here where, what was that? Say the last part?
When the air pressure is equal to the water pressure.
That's right, that's right. So it's going to move in, and so this water has pressure, which is why it's moving in. but when the air pressure is equal to that water pressure, it will stop. And so water may go into here and stop. All right, and so you have equalized pressure here. So then let's look at if these seeds are doing cellular respiration. What's going on? What kind of gas exchange is occurring? If these seeds are doing cellular respiration. What are they taking out of the air in the responder? Cellular respiration, when gas goes in. Oxygen, right? So therefore, I'm going to write here O2. There's O2 molecules in here, the oxygen molecules. And so there's a bunch of oxygen in here. So there's some O2 molecules. Those molecules, that O2 is going to go in and be used by the seed for cellular respiration. What molecule does it pump back out at the end of cellular respiration? CO2, right? And CO2 though, when the CO2 comes out here, the CO2 is actually, instead of becoming part of the air in here, the CO2, this is the point of the KOH. The CO2, instead of coming out here, it's going to actually react with the KOH. So the CO2 is not going to be in this air here. It's going to react with the KOH. And when it does that, it forms a white solid. Potassium carbonate. And so at the end of the experiment, sometimes when you take that cotton ball out, you can actually see the white solid on it. So what happens here is as these seeds are going through cellular respiration, they're going to take in oxygen, but instead of replacing it with CO2, the CO2 is going to react with the KOH and be part of this cotton ball here. So therefore, what's going to happen to the number of gas molecules in here? Is it going to increase, decrease, or stay the same? It's going to decrease. And you notice the ratio up here with the equation? 6 molecules of O2 go in and 6 molecules of CO2 go out. So for every 6 that go in, you're not replacing it with the 6 CO2s. The 6 CO2s are going to react with the KOH, and so you're going to get less and less gas molecules here. If you get less and less gas molecules inside the respirometer, will that affect where the water is? Do you think? Do you have less gas inside of here? Absolutely, and so the water will start to move in. All right, so let's look at why that is. On the 3rd page of your sheet, blast from chemistry, remember ideal gas law? PV equals nRT. All right, so I'm going to just do a quick little review about this and how it relates to this lab. So ideal gas law. So it tells you what all those things, the PV and RT, what they are. I have 3 little questions here that I'd like you to answer using the ideal gas law. So if pressure increases at a constant temperature and N, what happens to the volume? So see if you can answer these 3 questions. Remember these, or you want me to go through the like example, like the first one? First one? I appreciate it. Somebody else is doing that. You guys ready to go over these? Yeah. All right, so let's look at, I'm just going to write it next to it here, PV equals nRT. All right, so if pressure increases, so it's telling you pressure is, I'm just going to write underneath it here. So pressure is increasing. And it tells you you have a constant temperature in N. What's N? Number of the gas molecules. So I'm just going to underline the things that are staying the same. So N and T are constant, so those aren't changing. What's R? A gas constant. R is never going to change, so R is always going to be a constant. And so this side here is staying the same, and pressure is increasing. So therefore, to have this side equal this side and nothing is changing on this side here, what must happen to the volume? The volume decreases. All right, so you can see here if I had a certain number of gas molecules in a space, and I had a plunger here, and so here's your gas molecules. You have a number at a constant temperature, not adding or taking away any of the gas molecules. If I increase the pressure by pushing down the plunger, all right, then it's going to squish those gas molecules closer, and our volume will decrease. Okay. All right. So then number 2, if temperature increases at a constant volume and N, what happens to P? So PV equals nRT. All right, we have temperature increasing at a constant volume and N.
Excuse interruption for a few announcements. The CHS Optimist Club will be meeting today due to no school tomorrow. So the meeting is in room 218 in room 220. In room 218 at 225. Also remind the students that there is no school tomorrow, so we'll see you on Wednesday. We have one bus change, bus 53 will be in for 18. And now here's Mr. Schwab with the results of the political poll.
Good afternoon, CHS. You have spoken, and the following are the results of our school-wide election held today. We had a record voter turnout this year with 656 students casting their ballot, so thank you. Coming in 4th, representing the Green Party, is Jill Stein. The next runner-up, representing the Libertarian Party, was Gary Johnson, with less than 4% of the vote. According to the student body at CHS, the next president of the United States, representing the Republican Party, is Donald Trump with 65% of the vote. And coming in at second, carrying the Democratic banner, was Hillary Clinton with 24%. Thanks, CHS.
All right, hold on, you guys, real quickly. Let's finish this one. So, if the temperature increases at a constant volume, N and R, what happens to P? So if this increases, what must happen to the pressure? Increase as well. All right, and then the last one, PV equals nRT. N is increasing at constant temperature, and R is always constant here. What must happen to this side altogether? It has to go up. How could this go up? One or both of these need to increase. Does that make sense? All right, so tomorrow, or no, not tomorrow, Wednesday, we will relate this to the drawing with the lab. Hold on, guys. On Wednesday, you guys are going to design your own experiment in groups. So I'm pushing everything back one day because you have to design and you need we're going to need the class time to do that on Wednesday. So we're going to actually do the lab on Thursday, wrap up on Friday, test on Monday. All right? All right. Test Monday. All right? All right?